The difference between a prokaryotic and a eukaryotic cell involves the internal structure of the cell. Prokaryotic cells, remember, are single-celled organisms. So they're organisms that are made out of one and only one cell. These types of cells have no organelles. They have no internal membrane-bound compartments that we call organelles. In comparison, eukaryotic cells have organelles. In terms of the domains of living things, both the bacteria and the archaea are prokaryotic. They are single-celled organisms, and their cells have no organelles inside of them. The prokaryotes on our planet vastly outnumber the eukaryotic organisms. There are just many, 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 many more species of prokaryotic life form than there are eukaryotic life forms. Prokaryotic life forms also occupy a wider range of environmental niches than any other organism. So they're just able to be found in many more locations in the environment around the globe than eukaryotic organisms are. Fewer than 1% of the bacterial prokaryotic life forms on our planet are harmful to us. That's an important thing to know. We sometimes imagine that bacteria are organisms that cause problems for us, but in reality, the vast majority of bacterial organisms do not do us harm. In fact, many of them are quite helpful to us. They live on us and in us as part of what's called our microbiome. So this number, 1%, this will vary a little bit depending on the source that you're reading. It can go as high as 5%, but only 1 to perhaps 5% of all of the different kinds of bacteria are potentially harmful to us. The rest of them are not. The other thing that you should know is that there are currently no archaeal species that are known to cause disease in humans. There's not a single species of archaea that is known to cause a problem to human health. The other interesting thing to know about archaea is that, like the bacteria, they occupy a lot of different environmental spaces or niches around the planet. But the archaea in particular tend to occupy really extreme places in our world. For example, archaea can be found in some hot springs where all other types of cells would die from the heat. Archaea can survive in particularly cold places. They can survive in extremes of pH. They can survive in places that are much saltier than other organisms can survive. So the archaea have evolved to take advantage of locations in the environment where other things just can't survive. They're an interesting group of organisms for sure. We said that prokaryotic cells vary in their shape. They don't vary as much as eukaryotic cells do, but they do vary in their shape. Where you really see diversity among prokaryotic cells, though, is in their external structures. So the kind of structural components that exist outside of the cell membrane. Remember, all cells have a certain list of things inside of them. All cells. All cells have a membrane, they all have cytoplasm, they all have DNA, and they have ribosomes. Prokaryotic cells really stop there. Eukaryotic cells also contain organelles and a true cytoskeleton. But prokaryotic cells are much more basic in their internal structural components. Where they differ is in what's happening outside of the membrane. And that's what we'll take a look at today. On this slide, you can see a drawing of a prokaryotic cell. This would be considered a rod-shaped cell or a bacillus-shaped cell. And you can see that the cell has been cut open, so we can see inside. The blue line that you see traveling around the edge of the cell, that represents the cell membrane. If this is a bacterial cell, you can see that it's got this pink-purple layer outside of its cell membrane called the cell wall. Remember, bacteria have a cell wall made out of that complex carbohydrate, that polysaccharide called peptidoglycan. It's the only place we find peptidoglycan, in the cell wall of bacteria. Inside of this cell, we can see this sort of mass of what looks like spaghetti. This is the nucleoid. That's the DNA molecule that has been wound around protein and then carefully stacked together into this structure called a nucleoid. Remember, this is not a nucleus. Prokaryotic cells don't have a nucleus. So their genetic material is actually floating in the cytoplasm, although it is folded up. This yellowish gold material, this represents cytoplasm, and these bluish circles represent the ribosomes. Now there's another structure visible in this drawing, and that's right here. There's sort of a yellow circular structure here, and there's another one over here. These are called inclusion bodies. These are not organelles. There's no membrane around this, but they are specialized storage compartments in prokaryotic cells, and we'll talk more about them in a few minutes. Now, if you go to the outside of this cell, we can see over here a long, thick structure like a tail called a flagellum. You can also see these hair-like structures coming out on the exterior side of the cell. Like eukaryotic cells, prokaryotic cells have these tails, these flagella. But remember, we said that eukaryotic cells have cilia, or hair-like structures on their exterior surface. Prokaryotic cells don't have cilia, but they do have hair-like structures sometimes. In fact, they have two different varieties of these. One is called pili, and one is called fimbria. These that are drawn in this image represent fimbria. When we take the nucleoid out of a prokaryotic cell and we remove all of the histone-like proteins that it's wrapped around, the DNA has a very unique look to it. In most prokaryotic cells, the DNA molecule is circular. There is a single circular chromosome or piece of DNA inside of most prokaryotic cells. There are some exceptions. In fact, it was only very recently that a couple of species of bacteria were discovered that have two circular chromosomes, but most of them only have one. So unlike human beings, for example, who have linear pieces of DNA, prokaryotes have circular DNA. And most of their DNA is found in this one circular piece. All of the genes that are necessary for the survival of the cell are found in that circular chromosome. Now, what's interesting is that in some prokaryotic cells, there is additional DNA to be found. There are much smaller circular pieces of DNA called plasmids. Not all prokaryotic cells have plasmids in them, but some do. Some, in fact, have multiple plasmids in them. Plasmid DNA is considered extra chromosomal. In other words, it's outside of the chromosome of the organism. That means that the genes that are found on plasmid DNA are not necessary for survival. They are sort of additional genes, and they tend to code for proteins that give the cell an additional useful trait. For example, the plasmid DNA found in some bacteria contains the genes that are necessary for antibiotic resistance. Bacteria that have these plasmids inside of them are able to resist or survive being exposed to certain antibiotics. So that DNA, that plasmid DNA, is giving that bacterial cell a survival advantage. It's giving it an extra trait. It's a trait that most other bacteria don't have. But that particular bacteria that has that plasmid now has that special trait, the ability to resist antibiotics. One interesting thing to note about bacteria and other prokaryotic organisms like archaea is that plasmid DNA can be transferred horizontally, in other words, from one cell to another, from neighbor to neighbor, in other words. Plasmid DNA can be transferred around the neighborhood from one cell to another. That's one of the ways, for example, that antibiotic resistance gets transferred from one type of bacteria to another, is through this horizontal transfer from cell to cell. If you take the circular chromosome out of a prokaryotic cell, the primary genetic material, the chromosome or the piece of DNA that has all of the genes in it that the cell needs to survive, if you take that DNA and you examine it, it's much simpler than what we see in eukaryotic cells. Now, what I mean by simpler is, first of all, it has many fewer base pairs in it. Remember, the DNA molecule is double-stranded, and the bases, the A's and the T's and the G's and the C's, the bases that stick off of it are going to pair with each other in a complementary way, A with T and G with C. So everywhere there's an A hydrogen bonded to a T, that's a base pair. Everywhere there's a C hydrogen bonded to a G, that's a base pair. Bacterial DNA has many fewer base pairs in it compared to human DNA. And that means that it's just a shorter molecule and it has fewer genes in it. For example, in E. coli, E stands for Escherichia. Escherichia coli, that's a species of bacteria. The genus name is Escherichia, and the species name is coli. This particular kind of bacteria has 4.6 million base pairs in its DNA, about 4,300 genes. Now, if you compare that to human DNA, we've got about 3 billion base pairs, billion with a B, and approximately 30,000 genes. So, the DNA molecule in prokaryotic cells is just simpler. It's smaller, It has fewer genes in it. That makes sense because prokaryotic organisms are just simpler organisms. They are not nearly as structurally and functionally complex as a human being is. On this slide, you're looking at an electron microscopy image of an E. coli. Now, this is a TEM image, TEM, transmission electron microscopy. Remember, we said that type of EM is going to show us the internal details of the cell. This material right here in the center, this is the nucleoid. This is the DNA all wrapped up and folded in with histone-like proteins. If you go out to the edge, you can just see two layers. The innermost layer is the membrane, and then beyond that is the cell wall. The rest of the cell contains cytoplasm and these black dots. These are ribosomes. This cell is just packed with ribosomes. And remember what ribosomes do. Ribosomes are the cellular machines that translate RNA and make protein. Let's take a few minutes and talk in a little more detail about these ribosomes. On this slide, on the right-hand side up top, you're looking at another TEM image of a bacterial cell. Now we're seeing this cell lengthwise, whereas we saw the previous cell in cross-section or transverse section. But we can see the same structures in here that we saw in the last image. We can see the cell membrane in red and the cell wall in green. Those have been colorized, by the way. There is no actual color in those structures. This represents the nucleoid. This is the DNA and the histone-like proteins. And then you can see all of these ribosomes in here. Amazing. Now, it's hard to see, but there's another layer outside of the cell wall in this EM image. It's very light. It's colored a very light yellow. But this particular species has what's referred to as a capsule around it. That's one of those extra external structures that we said many different bacteria have. It's some of the diversity in structure that we see in prokaryotic cells. We'll talk more about capsules in a few minutes. The ribosomes themselves are remarkably similar between prokaryotic cells and eukaryotic cells. All ribosomes are made out of two subunits, one large subunit and one small subunit. Those two pieces come together and function together as the ribosome. The junction between the two subunits is where the actual translation process occurs. In other words, the messenger RNA molecule is going to come in right here. It's going to fit right here between the two subunits. The ribosome is going to read it and translate it. It's going to translate the code that's in the RNA, and it's going to build a protein from it. So it doesn't matter if you're looking at a prokaryotic cell or a eukaryotic cell. Both of these subunits, the large subunit and the small subunit, both subunits are going to be made out of rRNA and protein. Ribosomal RNA and protein. Those two types of macromolecule are all wound up together in the large subunit and in the small subunit. Now, when you look at a prokaryotic ribosome and you compare it to a eukaryotic ribosome, they look the same and they function the same. but there are some differences between them, and those differences come in the exact type of protein and the exact type of ribosomal RNA. In other words, the order of the bases in the rRNA, the length of the piece of rRNA is going to differ between a prokaryotic cell and a eukaryotic cell. They all contain rRNA and protein, but the exact protein, the exact rRNA is going to differ. That's what they're showing us down here in the image. Don't worry about these numbers. It's nothing you need to know for this course. Just understand that it's not that the ribosomes differ in their composition. They all contain protein and rRNA, but prokaryotic cells have different rRNA and protein than eukaryotic cells do. The other thing that you can see between prokaryotes and eukaryotes in terms of the ribosome is the measurement of the ribosome. Now, I didn't say length or diameter or anything like that, because the way we measure ribosomes is with this unusual unit called a Svedberg unit. And a Svedberg unit is shown with a capital S. So prokaryotic ribosomes are 70 Svedberg units. Again, it's not length, it's not width or height or mass or anything like that. It's its own unit of measurement. The small subunit is 30S. The large subunit is 50S. Now you would think that that means that the whole thing is 80S, but it's not, because that's not how Svedberg units work. The whole ribosome is 70S inside of a prokaryotic cell. So prokaryotic cells have some slight differences in their ribosomes compared to eukaryotic cells. Prokaryotic cells also have structures that eukaryotic cells just don't have. One thing that prokaryotic cells have that eukaryotic cells don't have is a cell wall. And as we said, it's made out of that special polysaccharide carbohydrate called peptidoglycan. This cell wall is critically important to bacterial cells because that's the type of prokaryote that has a cell wall, bacteria. What it's going to do for the bacterial cell is give it protection. If you remember when we were talking about carbohydrates, we said that peptidoglycan is unique because it gives extraordinary strength to the bacterial cell wall. It allows that wall to resist all different kinds of forces, including pushing and pulling and twisting or torquing. The wall lies outside of or exterior to the cell membrane. Only bacteria have a peptidoglycan cell wall. The archaea have something else, and we'll talk about that in a minute. What's interesting is that the cell wall differs a little bit between different species of bacteria. Some bacterial species have a very thick cell wall. Other species have a very thin cell wall. It turns out that we can classify bacteria, we can help identify bacteria according to the thickness of their peptidoglycan cell wall. And we can find out how thick their cell wall is by staining it in the laboratory. We use a procedure called gram staining to do this. When you gram stain bacteria, if they have a thick cell wall, they're going to appear purple under the microscope. And we're going to call them gram positive. If they have a thin cell wall, it's going to appear pink under the microscope, and we're going to call them gram-negative. So among all of the many different types of bacteria in the world, the first way we can sort of start to differentiate them or group them together is according to their gram staining results, or what we call their gram status. Take a look at this drawing down at the bottom. This is an artist's representation of the difference between a gram-positive and a gram-negative cell. Over on the right here, we can see a little piece of the cell membrane right here. So we've cut a little bit off of this bacterial cell. Like all membrane, you can see the heads on either side. You can see the tails tucked in the middle. You can even see a piece of protein embedded in the membrane. These are things that are found in all cell membranes. Now, this area over here represents the inside area of the cell, and this represents the outside. So here's the cell membrane, and here is the cell wall. These represent strands of peptidoglycan, and as you can see, there are several of them. This is a thick layer of peptidoglycan. This is a gram-positive species of bacteria. After it was gram stained, when we looked at it under the microscope, it would appear purple. Now compare that to this one over here. We've got a different species here. It's got that cell membrane because all cells have a cell membrane. But outside of that, it has a very thin layer of peptidoglycan. This is a cell that would stain pink after gram staining. This is a gram-negative type of cell. Now notice there's another structure here outside of the cell wall. We have a second membrane here. This is not the cell membrane. This is the cell membrane. This is an external or an outer membrane, and it's found in gram-negative bacteria. You can see that it's got some structural components in it that look different from a traditional cell membrane. On this slide, we're looking at a couple of images that were taken underneath a light microscope, showing us the difference between gram-positive and gram-negative cells. On the top here, you can see bacterial cells that are stained purple. These cells were put onto a glass microscope slide, and then they were put through the gram staining procedure, and this is what they looked like afterwards. Now, this image was taken with that high power oil immersion lens on the light microscope. That is the highest or the strongest lens on a typical light microscope, and it can magnify 100 times. Remember, the eyepieces also magnify a little bit, so this would be a total magnification of 1,000 times. Each little dot here is a cell. So you can see these cells, right? But you don't see any detail about them other than their shape. This is what we're limited to when we use a light microscope to visualize prokaryotic cells. We can see them, we can see their shape, but we can't get much other detail. Gram staining gives us a little more detail in that it tells us that these cells are gram-positive. So these cells must have a thick layer of peptidoglycan in their cell wall. They're also circular or spherical in 3 dimensions. So these would be called coccus-shaped. And the plural of coccus is cocci. So we would call this a gram-positive coccus. And this is actually an organism called Staphylococcus. You've probably heard of Staphylococcus bacteria. The genus name is Staphylococcus. And of course, we always capitalize the name of an organism's genus. Down here, we see something very different. Down here, we see pink cells. And these cells are not spherical. These cells are rod-shaped. Individual elongated cells. You can see one cell here, next to another, 3 different rod-shaped cells right here. This organism is gram-negative because it stains pink. And while we can see these rods a little more clearly than we could see the purple cocci in the other image, you still can't get much detail about these cells. You can see them, you can see their shape, but you can't see a lot of detail beyond that. This happens to be an organism called Bacillus. The genus name is Bacillus. So, most of the diversity between different species of prokaryotic organisms, so different kinds of bacteria and different kinds of archaea, most of the diversity is in what's outside of their membrane. So let's spend the next few minutes talking about those external structures. All bacteria have a cell wall. All archaea have a similar structure outside of their membrane, but it's not made out of peptidoglycan. So we don't call it a cell wall, we call it the S layer. So bacteria have a cell wall made out of peptidoglycan. Archaea don't have a cell wall made out of peptidoglycan. They have an S layer that replaces their cell wall. Outside of the cell wall, or the S layer, we sometimes see an additional layer called a glycocalyx. Now, there are 2 types of glycocalyces that you can see. One is called a capsule, and one is called a slime layer. They're both called glycocalyx, but they're different from each other in that a capsule is a layer that's very tightly organized, and a slime layer is quite loose, as the name would suggest. The reason that a prokaryotic cell would have a glycocalyx is because it helps the cell adhere to surfaces in its environment, and it also helps the cell evade detection by the immune system. Now, in terms of the S layer, we said that archaea have an S layer that serves as their cell wall, but you will see an S layer in some bacteria in addition to their peptidoglycan cell wall. So some bacteria have multiple layers of materials outside of their cell membrane. Both the glycocalyx, the capsule or the slime layer, and the S layer are made out of a combination of protein and carbohydrate. Remember, the cell wall in bacteria is made out of peptidoglycan. On this slide, you're looking at a couple of microscope images showing the glycocalyx. Let's start over here on the left. This image was taken with a kind of microscope that we didn't talk about, called a fluorescent microscope. As the name suggests, that type of microscope uses the property of fluorescence in order to see certain structures in or around cells. Fluorescent microscopy uses a special kind of staining using fluorescent dyes. That's what you're looking at here. Where you see black in here, you're looking at a group of bacterial cells, 1, 2, 3, 4 of them, lined up end to end. This black is the background. And then you've got this bright fluorescent green material here. This is glycocalyx. So this is material that sits outside of the cell wall of these bacteria. It's made out of a combination of protein and carbohydrate. It's called a capsule because it's very tightly organized. It's very tightly put together around the cell. Compare that to what you're seeing in this image. This is an electron microscope image, a TEM image of a piece of intestinal wall with some bacteria attached to it. So all of this over here, this is intestinal cell wall. Where you see white over here, this is the space in the cavity of the intestine. And then here and here and here and here and here, you're looking at bacterial cells. See this material that surrounds them, the bacterial cells? It looks like speckled, sort of spidery black lines inside of white material. This is slime layer. Slime layer is just much less organized. It's not adhered nice and tightly the way capsule is. It's still made out of protein and carbohydrate. It's just sort of looser and more slimy. Both of these types of glycocalyx are going to help cells adhere to surfaces. You can imagine that's very important for a cell that lives in the intestine. Adhere to surfaces and hide out from the immune system. Now, on this slide, down at the bottom, you're looking at an archaeal cell, and you're looking at the outside of its S layer. So this is an EM image, but instead of being transmission electron microscopy, this is scanning electron microscopy. Look at the difference. You can't see into this cell. All you can see is its surface. So we're looking at the surface of the S layer, the material that replaces the cell wall. Notice that it has this very regular pattern on it. To my eyes, it almost looks like a bandage. It looks like it has the texture of a bandage, doesn't it? Over here at the top, you're looking at a TEM image, so we can see internal details of this bacterial cell. And what's marked CW is the actual cell wall made out of peptidoglycan. In addition to the layers that can surround the cell membrane, there are also these appendages that we see outside of prokaryotic cells sometimes. These external appendages are involved in helping the cell move through its environment and helping it attach to surfaces. Now, we mentioned the flagella. That's the tail-like structure that you see in some bacterial and archaeal cells. You can also see flagella, by the way, in eukaryotic cells. Now, the other two appendages are pili, that's plural, pilus is singular, and fimbriae. Fimbriae is plural, fimbria is singular. Now, if you compare these, they're all elongated linear structures. The flagellum is the largest and the longest. Cells can have one or several flagella attached to them. Next longest is the pilus. The pilus is shorter than the flagellum, but longer than the fimbria. And finally, you have fimbria, which is the shortest and most thin type of appendage, very hair-like. It's the fimbria in prokaryotic cells that are similar to cilia in eukaryotic cells. In these images down at the bottom, if we start over here on the right, these are cells taken with a scanning electron microscope, SEM. There's 2 cells here. They're head to tail. The longest of these structures that you see, these are flagella. The next longest, remember, are called pili, and then the shortest are called fimbria, and you can see fimbria on these cells. So these cells have both flagella and fimbria on them. That's not uncommon. This image right here was taken with a fluorescent microscope. Here's one cell, here's another. And what you're seeing stained red here, these are pili. Longer than fimbria, but shorter than flagella. I want to spend the next couple minutes talking about a couple of the differences that we see in the cell membrane between prokaryotic cells and eukaryotic cells. Remember, all cells have a plasma membrane or a cell membrane. And all plasma membranes have hydrophilic heads and hydrophobic tails. So the heads are going to be anywhere that faces the water, and the tails are going to be tucked in the middle. All cells have this arrangement. What's interesting in the prokaryotes, though, is this. The structure of the tails actually differs between bacteria and archaea. Just like us, the bacteria have 2 fatty acids in each of the phospholipid molecules that makes up the membrane. Archaea are different. Archaea don't have fatty acids as the tails in their membrane. They use a different molecule. It's a molecule called isoprene. It's a much more ancient molecule, evolutionarily speaking, but it has enough similarity in its chemistry to fatty acids that it can replace them. Isoprenes are hydrophobic, so they will behave the same way that fatty acids behave in the membrane. The other thing to note is that prokaryotic organisms don't have cholesterol in their membranes. Only animal cells use the sterol called cholesterol. Plant cells use a different kind of sterol. The prokaryotic organisms, they actually use what's called hapanoids. There's a molecule called a hapanoid that these types of organisms substitute for the more modern type of sterol that we see in other cells. Like cholesterol, a hapanoid has a 5-ring structure. So we saw how cholesterol has a very bulky ring-like or ring-based structure. Hapanoids also have a ring structure, but they're not considered sterols because they're missing that little hydroxyl group at one end that we mentioned. So hapanoids are sort of the pre-sterol molecule. On this slide, you're looking at a drawing of the chemical structure of a hapanoid next to the chemical structure of a sterol. All right, you can see a lot of similarity. The cholesterol has 3 6-carbon rings and 1 5-carbon ring, and it's got a hydroxyl group. The hapanoid's a little different. It's got 5 rings, it's got an additional 6-carbon ring, and it has no hydroxyl group. So it's not a sterol, technically speaking, it's a pre-sterol, but it substitutes in prokaryotic cells in their membrane for what sterols do in other types of cells. Now, when it comes to the functions of the cell membrane, it doesn't matter if you're looking at a prokaryotic cell or a eukaryotic cell. The membrane is going to act as a permeability barrier. It's semi-permeable. And of course, it's loaded with protein. All types of cells are full of membrane proteins. But here's a big difference between prokaryotic cells and eukaryotic cells. Remember, a eukaryotic cell is going to make its ATP in the organelle called the mitochondria. What prokaryotic cells do is different. They don't have mitochondria. So they still need ATP. All cells need ATP, but they have to make it in a different location. And that location is the plasma membrane. They use their plasma membrane to make ATP instead of the mitochondrial membrane. The last thing I want to talk about in this lecture today are those inclusion bodies that we sometimes see inside of prokaryotic cells. Remember, inclusion bodies are not organelles. They don't have any membrane around them, but they do function as storage compartments. Now, the kinds of things that you see stored in inclusion bodies include things like fats for energy reserves, carbon for the synthesis of all different types of molecules. Various minerals can be stored in inclusion bodies. And here's the most interesting one. There's a type of inclusion body called a magnetosome. And like the name suggests, inside of a magnetosome inclusion body, there are magnets. There are little tiny cellular magnets. And you might ask yourself, why in the world would a cell store magnets? Well, the answer is to help itself orient where it is in the environment. Some prokaryotic cells use magnetosomes in order to line themselves up with the electromagnetic fields of the Earth. It's a way for them to orient themselves as to where they are in their environment. Fascinating. Inclusion bodies are visible with a light microscope, so let's take a look. On this slide, you're looking at two EM images. This is a transmission EM image of a bacterial cell. Inside this cell, you see something very unusual. You see all these big white circles or ovals of material. These are inclusion bodies, and they are loaded with fat. That's what this is. This particular bacterial cell, for some reason, is storing a whole lot of fat. And you would store fat for future energy needs. Over here in the other image, we're looking at magnetosomes. Again, this is a TEM image, transmission electron microscope. We can see nucleoid in here. We can see ribosomes, the black dots in here. And then we see this thing. This is an inclusion body that we would call a magnetosome. And it's got these circular black bits of material in here. These are literally magnets. And inside this inclusion body, they're going to line up and orient the cell to its position in the environment using the Earth's magnetic fields.